Hello and welcome to my latest blog. Today we're going to talk about non-tuberculous mycobacteria and the Mycobacterium tuberculosis complex, bad bugs hiding behind the main villain. Tuberculosis is a disease we've probably all heard of, and in England, where I grew up, and much of the global north, tuberculosis, or TB, is often thought of as a historical disease such as plague or smallpox, and conjures up images of swooning Victorian women and sanatoriums. But if you come from the global south, or even Eastern Europe, you'll know that TB is definitely still a disease of now. You can read more about the history and current day problems we're facing with TB on the blog. Historically, scientists thought that TB was caused by a single species, called Mycobacterium tuberculosis. More recently, however, we've begun to realise that tuberculosis, the disease, is actually caused by a number of different mycobacterial species. These species are often found in the environment or living in other animal hosts, so humans are often accidentally infected. M. tuberculosis, on the other hand, and despite much research, does not appear to survive in the environment or infect other animals, making us the obligate or only host. There are two main groups of mycobacteria that can cause TB-like disease. Often, and slightly confusingly, the strains within these groups often have their own complexes of substrains, which may also change the course of disease and treatment. These two groups are the Mycobacterium tuberculosis complex, or MTC. Mycobacterium tuberculosis is of course the celebrity in this family of organisms, but there are lots of genetically very similar species that we now know can also cause TB. It's really only since the development and the use of sequencing that we've been able to distinguish between them, and phenotypically, when you compare how they look, or also called morphology, they're often indistinguishable. In the laboratory, they have varying levels of risk. Some are classified as hazard group 3 pathogens, which are highly contagious and very hard to treat, and the same level as M. tuberculosis, and they must be worked on only in special biological safety level 3 laboratories. Others are not thought to be so dangerous and are classified as hazard group 2 pathogens. They may cause disease, but they're not quite so contagious, or the route of transmission may be less dangerous. Some of the MTC species may be more familiar, and this obviously depends where you live in the world. But as the name suggests, Mycobacterium bovis primarily infects cows, and it has been a problem in countries like the UK for decades. It causes TB-like disease in cows and also other mammals such as the badger and humans, and there have been ongoing debates as to whether culling badgers may stop the spread of M. bovis. It's caused farmers much distress, both financial and psychological, with many herds having to be destroyed due to outbreaks. M. bovis has also given rise to the only vaccine that we have for TB so far, the Bacillus calmette-guerin, or BCG, strain of M. bovis. The vaccine is a live, attenuated, or weakened version of the original fully pathogenic M. bovis, and this stimulates the immune system into thinking it's being attacked by a TB-causing organism. The history of the BCG vaccine is really interesting, and we recently whole genome sequenced a large collection of BCG strains that we'd had in our laboratory for some time. And you can read the plain language summary of our BCG sequencing paper and more about BCG in general in the link on my blog. Some other interesting species include Mycobacterium africanum, which, as the name suggests, circulates mostly in West Africa, and it's thought to cause up to 40% of all of the TB cases in the region. Like M. tuberculosis and M. conneti, which is another MTC, humans appear to be the only host for M. africanum. A slightly more unusual member of the MTC is M. pinnipedi. Named after the hosts it usually infects, the pinnipeds, it's most commonly a pathogen of seals. There have been reports of this opportunistic zoonotic bacterium infecting humans who have close contact with seals and similar species, notably in zookeepers. Now, the other group of TB-causing or TB-like causing organisms are the non-tuberculous mycobacteria, or NTMs. They're more distantly related to M. tuberculosis than MTC, and they cause often slightly different disease symptoms, although sometimes can be indistinguishable from those caused by M. tuberculosis. They're often opportunistic human pathogens and produce non-tuberculous symptoms, often with less pathogenic power, although many are really hard to treat and they can hang around, especially in immunocompromised patients. They often cause lung diseases in people with already diseased or damaged lungs. According to the charity Asthma and Lung UK, NTM infections in the UK are rare, with approximately 7.5 cases per 100,000 people. It doesn't always cause disease though, and some people can carry the bacteria around in their airways without them causing any problems, so it doesn't necessarily mean you'll need treatment, which can be long and very difficult, though it does mean you may potentially be transmitting it to other vulnerable people. The NTMs are differentiated from the MTC, as whilst they do cause TB-like pulmonary lung disease, they technically don't cause tuberculosis. It's actually called mycobacteriosis instead, though they are very often misdiagnosed as TB to begin with. They occur in humans and animals, and they can also be found in the environment, especially soil and water. The three most common NTM species are M. abscessus, the M. avium complex, and M. kansasi. M. abscessus is particularly interesting as it can not only cause lung disease, but can infect almost any organ in humans, and particularly causes skin and soft tissue diseases. It's commonly found in soil and water and is becoming increasingly familiar to doctors of immunocompromised patients, as it's extremely difficult to treat due to the number of drug resistances it intrinsically possesses. There are other pathogenic mycobacteria too, and whilst they don't belong to either of the above pathogenic mycobacterial groups, I do think I should mention M. leprae and M. leptomatosis, the causative agents of leprosy or Hansen's disease. The history of leprosy is fascinating, and it's interesting that it's often again thought of a historical disease such as plague, when in actual fact it still occurs in some areas of the world today. Why is it a big deal then that there are more than one species that cause TB-like disease? Because a lot of countries don't have the capability to differentiate the different mycobacterial species, we don't have much reliable data, but it's thought that around 30% of TB infections might actually be caused by the MTC or NTMs rather than mycobacterial tuberculosis. There are some molecular tests such as PCR, mass spectrometry, and line probe assays that can detect the different species, but they're not always available, especially in lower middle income countries where the MTC and NTMs are thought to be a particular problem. Because they don't just infect humans, but also other animals and can be found in the environment as well, it's going to make it much harder to eliminate TB-like disease, even if we could get a grip on M. tuberculosis, and we've struggled with that so far, despite humans being the only host. But what makes MTC and NTMs really problematic is the fact that they often have very different drug resistance profiles to M. tuberculosis, which means if you're treating a patient empirically, and many countries have a set standard first-line drugs that all TB patients are given until the resistance testing can be done, that set of drugs might not work if the patient has, for instance, M. abscessus. These NTM and MTC often have many more resistances as well, and often multi-drug resistant cases of TB are misdiagnosed and are actually an MTC or NTM. The infections resulting from these pathogens are even more difficult to treat due to their intrinsic resistance to antibiotics like penicillin, sulfonamides, tetracycline, erythromycin, and chloramphenicol.